very warm welcome to today's Cell and Gene Therapy Insights webinar titled Tolerant Endonucleases and the Benefits for Viral Vector Manufacturing and a Comparison of Two Marketed Enzymes. I'm Lauren Coyle, an editor at BioInsights, and joining me today is 𝗞𝗲𝘅 and Svenja Mikulec, who will introduce DENARASE High Salt, an engineered 𝗟𝗲𝘅 endonuclease optimized for higher salt tolerance to improve the efficiency of viral vector manufacturing. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. Now, I'd like to introduce our presenter. 𝗞𝗲𝘅, co-founder and managing director of c-LEcta, holds a PhD in biochemistry and has over 15 years of experience in industrial biotech, specializing in product development and strategic alliances. Svenja Mikulec, a product manager at c-LEcta overseeing the biopharma portfolio, including DENARASE High Salt, leverages her background in leukemia research to improve viral vector production for cell and gene therapies. So without further ado, I'll hand over to Mark to kick off today's presentation.
Thank you very much, Lauren. Also a very warm welcome from my end. I'll kick it off here with a short introduction of c-LEcta as a company, and then I'll quickly hand over to Svenja to speak about the main topic of our webinar today. c-LEcta is located in Leipzig, which is in the eastern part of Germany. We were founded back in 2004 as a spin-off from university. So this year we are celebrating our 20th anniversary. We currently employ 125 people, and we have a very strong focus on research and development. Around 65% of our resources are dedicated to innovation. Everything at c-LEcta is about enzymes. Our core business is the development, production, and commercialization of enzymes, and this mainly for food and pharma applications. And we are running a collaborative business model where we like to engage with partners in the industry to do joint product developments in open innovation initiatives. So if you have any ideas on like how enzymes can be used or where new enzyme solutions are missing, please reach out. We'll be very happy to discuss. If you look out for a new enzyme for a new application, you will find out that most of the enzymes you can find in nature will not be adapted to the need of that application you're looking for. So usually it's a set of properties that enzymes need to fulfill, and if you map these properties with natural enzymes, you'll find out that very often they don't fulfill all the parameters. And that's where enzyme engineering comes into play, a methodology with which you can improve the properties of an enzyme or even come up with completely new enzyme solutions. And we at c-LEcta, we have developed such a platform to do enzyme engineering over the last 20 years. And it's basically mimicking the principles of natural evolution to improve enzymes in a step-by-step cyclic process. And there are two very important elements when doing this. On the one hand, the enzyme variant library design and creation is very important. We apply bioinformatics, machine learning, artificial intelligence to design enzyme libraries that have very high hit rates so that the screening effort is not that huge. And we speak about smart libraries. And we invested a lot of effort into developing molecular tools to be able to produce these newly designed libraries in a very fast and efficient way. The other element, which is of course super important in enzyme engineering, is the screening part. Enzyme variants libraries need to be screened, and ideally with a high throughput. But of course, we also need to consider that the enzyme we develop needs to fulfill the requirements of the application. So that means usually that we need to look at a couple of properties. We are speaking about multi-parameter screens to really make sure that we screen for something that works out in the targeted application. And Svenja is very soon going to speak about one very nice example, an enzyme that we have developed with our platform. Since 2022, c-LEcta is part of the 𝗗𝗲 group. 𝗗𝗲 is a world leader in sustainable taste and nutrition. And we are also running a strong pharma business unit where we are selling excipients, coatings, and cell nutrition solutions to the pharma industry. And 𝗗𝗲 is also a significant player in the enzyme space. And here you can see, and that's my last slide, the enzyme technology portfolio that we have, enzyme solutions for the different end-use markets that we are addressing. The green ones are the one in the food sector, and here already several of these solutions were brought in and developed by c-LEcta. And down on the right, you can see in red our pharma enzyme portfolio, and that is also a portfolio that came in through the c-LEcta acquisition by 𝗗𝗲. And now I hand it over to Svenja, who will explain you more about our pharma solutions.
So thanks for the kind introduction, Lauren, and also a warm welcome from my side. In the following, I'd like to discuss with you by c-LEcta engineered a salt-tolerant version of our endonuclease DENARASE and how it compares to other enzymes on the market. Endonucleases, most of you might be using them, are enzymes that specifically hydrolyze the phosphodiester bonds between nucleotides, which basically means that they can effectively break down all forms of nucleic acids. They are commonly used to remove contaminating DNA arising during purification of viral vectors used for vector and conjugate vaccines, as well as cell and gene therapy applications. DNA resulting from lysis of genetically transformed, often immortalized cell lines that are typically used to produce viral vectors can potentially be infectious, immuno and/or oncogenic, and therefore pose a potential threat to patients. With significant progresses being made in the development of viral vector-based therapeutics, the FDA thus has set acceptable limits for the amount as well as size of any residual DNA in the final drug product. To meet these requirements, the wild-type endonuclease derived from a bacillus species called Serratia marcescens remains the industry standard for enzymatic DNA removal, as it degrades plasmid and host cell DNA to fragments that are around 3 to 5 base pairs inside without affecting viral particle integrity. Due to its very reliable performance, our GMP-grade Serratia marcescens endonuclease DENARASE is used globally by over 250 customers. And since its launch in 2012, DENARASE was qualified by most top pharma companies and is applied in several commercial as well as hundreds of clinical and preclinical manufacturing processes. The widespread use of endonucleases like DENARASE led to a rather standardized workflow in which, after viral vector expression in host cells, virus particles are harvested. This might require a lysis step depending on the virus type. It is accompanied or followed by endonuclease treatment before further purification steps. Efficient DNA removal, however, not only enhances the safety profile of viral vector-based therapeutics, but is also highly beneficial for the overall process efficiency. By reducing the viscosity of cell lysates, endonuclease treatment facilitates downstream processing, resulting in overall higher vector yields. However, we all know there is always the room and also the need for further improvements to, in the long run, make innovative therapies more safe and affordable. For quite some time now, high salt concentrations have been discussed as one tool to further enhance production yield and safety of certain viral vector types, such as AAV. It was shown that using salt concentrations between 200 and 400 millimolar can significantly improve the solubility of viral vectors by reducing formation of aggregates, especially at higher cell densities and vector concentrations. Additionally, high salt helps making the DNA more accessible for degradation, and this results in improved vector yield and stability, as well as higher purity and safety of the final drug product. So this is great. However, the approach does not come without challenges, since wild-type 𝗟𝗏 marcescens endonucleases like DENARASE are inhibited by increasing salt concentrations. And currently available salt-active nucleases exhibit rather low activity plus suboptimal salt and pH profiles. Therefore, today, production processes often still need to be adapted to the used endonuclease and not the other way around. To address this issue, we decided to take the challenge to engineer a salt-tolerant version of our DENARASE that ideally works under a new spectrum of process-relevant high salt conditions. To understand the ideal characteristics of an optimized endonuclease, we collected feedback from over 40 customers working with hundreds of different processes. And from this extensive interaction with viral vector manufacturers, we concluded that the new version of DENARASE actually should be highly active over a broad salt spectrum and at a pH of 7.4, which represents typical process conditions. Sparking our curiosity and also ambition, there was only one way to find out if these specifications could actually be realized. And so our R&D scientists went to work and performed their, to me, enzyme engineering magic by introducing a sophisticated combination of just a few amino acid substitutions. They generated a set of DENARASE mutants with improved activity at high salt when compared to the wild type shown here in orange. When we analyzed the DNA removal performance of these DENARASE mutants at different salt concentrations, we could confirm that they exhibit high activity from 0 to 500 millimolar salt. And this robust activity across the entire salt spectrum was not only observed under standard 𝗟𝗏 marcescens endonuclease release assay conditions at pH 8, but more importantly, also at the much more process-relevant pH 7.4. Based on a very thorough characterization, we finally chose variant 3, the mutant that had the most optimal activity profile, and it is truly living up to its new name, DENARASE High Salt, under which it is now available for purchase or for testing. To shortly summarize its key characteristics, DENARASE High Salt is an engineered version of DENARASE, so the wild-type 𝗟𝗏 marcescens endonuclease. Both enzymes are produced using the same bacillus strain as a production host and share multiple enzyme characteristics like molecular weight, temperature optimum, and pH range. They slightly differ in their isoelectric point, and DENARASE High Salt requires a little bit higher levels of the essential cofactor magnesium. So all in all, the few introduced modifications enable DENARASE High Salt to clear DNA over a broad range of bioprocessing relevant salt conditions, while at the same time remaining so similar to the original DENARASE that a comparable manufacturing process and compatibility with the DENARASE ELISA kit for quantifying residual enzyme was maintained. To now give you like a rule of thumb on when to use one or the other enzyme, we compared the effect of increasing sodium chloride concentrations on their activity using our standard endonuclease activity assay, as also performed in our quality control labs. Therefore, we are showing data generated at process-relevant pH 7.4 and also at QC-relevant pH 8 conditions. Depicted in berry, you can see that DENARASE High Salt retains good performance at elevated salt concentrations, while the wild-type DENARASE, shown in orange, loses activity with increasing salt levels. Both enzymes show robust activity at pH 7.4, which is even enhanced when working at pH 8. But when you'll be testing our enzymes in your processes, you'll notice that it is not really necessary to increase the pH from physiological conditions for efficient DNA removal using our DENARASE enzymes. With unmatched performance at low salt conditions, the standard DENARASE continues to be the most economical solution from 0 to around about 200 millimolar salt. At higher concentrations, we clearly recommend to use DENARASE High Salt because it has a strong performance at a broad salt and pH range. It will be the most flexible and cost-efficient choice for processes that benefit from salt addition. Combined, the DENARASE portfolio thus offers unmatched flexibility, allowing you to simply choose the enzyme that best fits your process conditions. We were, of course, really eager to know how DENARASE High Salt compares to other commercially available salt-active nucleases. And to find out, we did an internal comparison study and measured DNA removal activities of various salt-active enzymes at different salt concentrations, which are represented by darker shades of blue in the graph. Since we used our in-house activity assay, we normalized measured activities to those provided on the manufacturer's COAs to do a fair comparison. On the graph, enzyme activities are shown relative to DENARASE High Salt at 250 millimolar salt. And honestly, having seen this graph many, many times, I'm still amazed by the results. Compared to ArcticZymes, SunHQ, and MSun, 𝗗𝗲𝘀 saltonase, DENARASE High Salt demonstrated by far the highest DNA clearance activity at every salt concentration. Even though for time and clarity reasons, I haven't put the data here, this superior performance was not only observed at pH 7.4, but was also evident at pH 8. Interestingly, looking, for example, at the salt profiles of SunHQ, the activity of most salt-active nucleases seemed to be somewhat dependent on the presence of elevated sodium chloride concentrations, whereas our enzyme maintains a very stable activity profile from no to high salt, which basically means that working with DENARASE High Salt will, in contrast to other available options, not require any buffer adjustments or unnecessary salt additions, allowing you to fully utilize your bioreactor capacity. We, however, realize that in vitro activity assays can never fully reflect real-life viral vector processing. Given our limited expertise and available methods at c-LEcta to analyze DENARASE High Salt under actual process conditions, we therefore started to collaborate closely with Cell and Gene Therapy Catapult. They are a well-known UK-based CDMO with extensive experience in viral vector manufacturing and process development, so the perfect partner for us. In a benchmarking study, they analyzed how well DENARASE High Salt removes DNA from their proprietary AAV2 lysates and compared it to ArcticZymes SunHQ, which was the market leader for salt-active nucleases at the time the experiments were being performed. Of course, identical amounts of both enzymes were used for each and every experimental condition. DNA removal efficiencies were monitored at pH 7.4, either without salt addition or a final amount of 200, 350, and 500 millimolar sodium chloride. We decided to also include one experimental condition at pH 8 and 500 millimolar salt, representing optimal conditions for SunHQ. Analysis was done directly post-lysis of host cells, as well as after an affinity capture step referred to as post-capture in the following slides. The different experimental conditions will be depicted using the color code you can see here on the right, with no salt addition represented as gray dots and increasing salt concentrations represented by darker shades of blue. Before examining DNA removal, the researchers at Catapult first quantified the general effect of different salt conditions on viral vector yield via capsid ELISA for AAV2. Consistent with previous findings, the addition of salt dose-dependently increased viral particle titers when samples were treated with DENARASE High Salt, indicating that high salt levels actually improve AAV particle release and solubility. Addressing the, to me, I would say, moderately big elephant in the room, with SunHQ, there was an unexpected variance in results. These fluctuations might be explained by suboptimal process conditions, especially too low salt and/or pH level, but also other factors could influence this, such as magnesium, resulting in below ideal performance of SunHQ. Next, DNA clearance from AAV2 lysates was quantified using picogreen. It's a pretty common standard method for the unselective detection of double-stranded DNA, which allows for the quantification of both residual plasmid and host cell-derived DNA impurities. Looking at post-lysis results on the left graph, DENARASE High Salt demonstrated a remarkably efficient and also consistent DNA reduction, irrespective of salt and pH conditions. And looking at the right graph, even resulted in DNA levels below the quantification limit in post-capture material. Application of DENARASE High Salt consistently achieved around 10 times lower DNA levels when compared to SunHQ. SunHQ also achieved DNA removal, but with, again, much higher variability and also less efficiency, especially at lower salt concentrations. Since the amount of residual DNA in most post-capture samples was below the detection limit and thus could not really be quantified using picogreen, additionally, a much more sensitive qPCR-based method that was developed by Cell and Gene Therapy Catapult was used to specifically quantify host cell-derived DNA. Similar to previous results, everyone was quite amazed by how efficient and consistent DENARASE High Salt removes DNA from Catapult's AAV2 lysates. Prior and post-capture, host cell DNA was reduced to around about 1 nanogram per ml after application of DENARASE High Salt. This was irrespective of the salt and pH condition. So for efficient DNA removal by DENARASE High Salt, it really doesn't seem to matter if or how much salt is added and whether the pH is at 7.4 or 8. In contrast, much less efficient and again very variable DNA removal efficiency was observed in SunHQ runs in all conditions we tested. So from the data we got from our great collaborators, Cell and Gene Therapy Catapult, we can conclude that DENARASE High Salt removes DNA from AAV lysates with highest efficiency. It outperforms regarding the extent and reproducibility of DNA reduction, and it works really, really well under production-relevant pH and high salt. So in summary, being highly active at a broad spectrum of process-relevant conditions, we believe DENARASE High Salt is the industry-leading enzyme in terms of performance for DNA removal. It is truly tailored to fit your process and can be easily adapted into existing processes and be integrated in your preferred process conditions. And last but not least, since we only introduced a few mutations to the original enzyme, DENARASE High Salt can be detected and quantified with the standard DENARASE ELISA kit. So you will not need to qualify several kits when using our DENARASE enzymes. Together with the original DENARASE, which we consider to be the most cost-efficient GMP endonuclease for low-salt processes, we can provide you with the complete toolkit for DNA removal for viral vector manufacturing processes. And with that, I'd really like to thank you all for your attention and invite you to get in touch if you want to learn more about the DENARASE portfolio or want to test DENARASE High Salt in your processes. There is a lot more data we can share, and we can provide free samples of both DENARASE enzymes. If you are around, please visit us at our booth at the BPI in Boston coming up in September. And now, Mark and me, we are, of course, very happy to take any of your questions.
Thank you, Mark and Svenja, for that great presentation. So as Svenja mentioned, we are now going to start our Q&A. So please keep your questions coming in, and we'll get to as many as we can and answer any other by emails. So to kick us off then, Svenja, you briefly mentioned that DENARASE and DENARASE High Salt differ in their requirements of magnesium. Could you please specify how much magnesium should be used for one or the other enzyme?
Yeah, so magnesium is an essential cofactor for Serratia marcescens endonucleases, and typically 1 to 2 millimolar magnesium is sufficient when you work with the original DENARASE, but due to the increased salt concentrations, you'll need around 5 to 25 millimolar of magnesium for optimal activity of the DENARASE High Salt enzyme. And for initial testings, we would probably recommend to start with a concentration of maybe 15 millimolar magnesium.
Thank you. Our next question here is for Mark. How many amino acid substitutions were made to generate the DENARASE High Salt?
Yeah, in this case, the amount of amino acid substitutions was pretty small. So at the end, only 4 mutations were needed to shift that salt profile from the wild type enzyme to where we wanted it to be.
And Svenja, this next one here is for you. How compatible is the DENARASE ELISA kit with the DENARASE High Salt?
Yeah, so as mentioned, DENARASE High Salt only differs in a few amino acids from the wild type. So for this reason, the ELISA kit can be used for detection and also quantification of DENARASE High Salt. But DENARASE High Salt shows slightly different affinity for the antibodies being used. And the kit only contains a standard solution of the wild type enzyme. So therefore, it's necessary to multiply ELISA kit readings of DENARASE High Salt by a correction factor to obtain accurate protein concentration. So this factor was determined in an extensive study done by our contract manufacturer that also developed the ELISA kit. And alternatively, one can also prepare a DENARASE High Salt standard solution. In the future, we plan to set up the kit as it contains both the wild type and the high salt standard.
Fantastic, thank you so much. And Mark, this one is for you. Where is the production site of the DENARASE High Salt located?
Yeah, it's produced in Germany, so the country where also c-LEcta is located in. So it's made in Germany.
Wonderful, thank you. And Svenja, how is the DENARASE High Salt performance affected by the presence of different detergents?
So, I mean, this is a fair question since detergents are typically present in lysis buffers. And this is why we also thought about this and analyzed the effect of, for example, Triton and Tween 20 and Tween 80 on the activity of DENARASE High Salt. And we could not see a negative effect of all detergents on enzyme activity when used at typical conditions.
And Mark, are there any different quality grades of DENARASE High Salt available?
Yeah. So yes, when we launched the product a couple of months ago, we brought different package sizes for an R&D grade quality. So so the enzyme can be used for process development work. Or we are currently working on bringing out a GMP grade quality tool that we expect to be able to launch in the second quarter of next year. So that is ongoing work. We're developing towards that target, and believe that we can bring it out in Q2.
Amazing. And Svenja, how well does DENARASE High Salt perform at different temperatures?
So the temperature range for the wild-type DENARASE and DENARASE High Salt is very similar. So they have a temperature optimum or peak at 37 degrees. And between 20 and 50 degrees Celsius, both enzymes retain over 40% of activity. Of course, we have temperature curves available that we can share that are also present in our validation guides. So everyone can get in touch and see the data.
Amazing, thank you. Another one here for Mark. Are there any plans for further engineered versions of DENARASE?
I mean, currently, I would say we are happy with what we have. We are not aware of any gaps or process conditions that our current enzymes could not cover. But of course, if there are any ideas out there, like where a new or different type of enzyme would be needed, we would be very confident that we could develop it. So if there is an idea around that, very happy to get in touch and discuss that opportunity.
Fantastic. And I think we have time for one more question. So Svenja, how can the DENARASE enzymes be inactivated?
So, I mean, typically DENARASE is used in bioprocessing of viral vectors, where it is in principle being removed during downstream processes. So 99% of enzyme is gone. So no inactivation is required. And also overdigestion wouldn't be possible since viral DNA is protected by the capsids. Normally, enzyme removal for like the standard serratia-marcescens-derived endonucleases happens as it simply flow through multiple chromatography steps, and these are typical part of the viral vector downstream processing. So latest after polishing, the enzyme is gone, and so far we didn't see any difference when customer tested DENARASE High Salt. For other purposes, you could add EDTA to remove the magnesium from the system, but this, however, would be a reversible inhibition. Very high heat could also be done, or you could add like a proteinase to get rid of the enzyme.
Wonderful. So thank you, Mark and Svenja, for answering those questions, but that is all we have time for today. Any questions we didn't get to, we will reply to by email, and this webinar will be available on demand tomorrow. So look out for an email from us with the link. All that's left is to thank Mark and Svenja once more for a great presentation, and thank you to you, our audience, for listening. We hope you'll join us again soon.